Hi everyone, my name is Kiara Olson and I've been working with Dr. Fran Sandemeyer to study the effects of immunization on Mojave Desert tortoises. The purpose of this research is to explore the relative importance of the adaptive and innate immunological systems of Mojave Desert tortoises, or Gophorus agassizii. Why is this research important, you might ask? So Mojave Desert tortoises have lived in the Mojave Desert for at least hundreds of years. In the mid-1900s, they were commonly seen and easily recognized by the locals. However, today they're rarely seen and in some places have disappeared entirely. They've been officially classified as a threatened species by the Fish and Wildlife Service and are considered a vulnerable species by the International Union for Conservation of Nature. Despite the laws that protect them, their numbers are still in decline. They're susceptible to a wide range of pathogens. That doesn't really help either. One of the most prevalent of these pathogens being upper respiratory tract disease. URTD is a chronic infectious disease responsible for population declines across the entire range of desert tortoise. The infection is highly virulent in some populations, while chronic or even dormant in others. The mechanism for whether environmental or genetic responsible for this diversity is not well understood. So it's clear that we need to actually understand how their immune systems work in order to treat them and effectively conserve the species. Now it's time for an immunology crash course. So say as a human, you get a cut on your arm and a pathogen enters your bloodstream. Your innate immune system is going to encounter it first. It's your first line of defense. Your phagocytes and your other white blood cells that comprise your innate immune system have broad recognition, meaning that they will attempt to recognize anything that is foreign and get rid of it. If they get overwhelmed and can't get rid of the pathogen on their own, then they have to go to their parents or the lymph nodes. When you start actually feeling sick, once you get that runny nose or a sore throat, that's when your acquired or your adaptive immune system is actually kicking into gear. The lymph nodes are going to take your B2 lymphocytes that weren't able to get rid of the pathogen the first time around and basically pump out a bigger, badder version of it that is specific only to that pathogen. This is known as a memory cell. Memory cells have very specific recognition. They remain present in your bloodstream even after they get rid of the pathogen the first time around. So that way, the next time you encounter that exact same pathogen, your body can get rid of it before you even get sick again. So this figure on the left side is displaying that initial encounter, that long response. And then on the right side of the figure, we see that sharp spike. And this is representative of when your memory cells recognize the pathogen, replicate it, and eradicate, or replicate and then eradicate it before you even feel symptomatic. So this is the mammalian definition of immunity. And we're unsure if tortoises actually have this. So this is basically the groundwork for our research. Now some experimental background for you guys. So 12 years ago, 7 tortoises were immunized with ovalbumin, which is basically chicken egg white protein. It's a large, flashy protein that should be easily recognized as a foreign entity by the tortoise. Their antibody response took about 1 to 3 months to reach their peak levels, and the antibodies remained elevated for less than a year. So if you take a look at the figure on the right, you'll see that the tortoises only exhibited about a 7-fold increase in antibodies over the course of several months, whereas humans produce a 100 all the way up to a 1,000-fold increase in 3 to 5 days. So these results display that tortoises take considerably longer to produce much smaller results. And it also posed the question of if they produce memory cells. So fast forward to present day, we re-immunized the tortoises that were previously immunized a new group of tortoises, and then injected a naive group of tortoises just with saline as a control group. So now we have a group of tortoises that has been immunized twice, a group that's been immunized once, and a group that's never been immunized. We have a total of 21 tortoises in our study with 7 to 8 in each of the groups. We hypothesized that if Mojave Desert tortoises exhibit immunological memory, ovalbumin or ova immunized tortoises will have greater titer values compared to non-immunized tortoises, which is indicative of the volume and binding strength of antibodies able to recognize ova. We predicted that the group that was immunized twice will display the fastest and strongest response of the groups, and the group that has only been immunized once will have a slower, more moderate response. And the naive group shouldn't produce any response. We utilized an ELISA, or an enzyme-linked immunosorbent assay, to test this hypothesis. After some Excel magic, we were able to determine a titer value for each of the test subjects. The titer curve provides a quantitative measurement of the volume of antibodies produced, as well as a measurement of the stickiness or the effectiveness of the antibodies produced. The more sticky the antibodies, the more steep the slope of the titer curve. And the titer curve displayed on the left is one from one of our tortoises, Paiute. So after some statistical analyses, we found that the second immunization group displayed no statistical significance in time to peak antibody titers or the peak values themselves when compared to the first immunization group. This is easily displayed by the statistics at the bottom of the slide with those large p-values. And then you can also see it in the figure on the right. So their peak titer values were both around a 4-fold increase from pre-immunization, and they reached it at about the same time. If the data corresponded with our hypothesis, then the group that was immunized twice should have exhibited much higher peak values than the group that was only immunized once, and should have reached these values significantly faster. So these results support the idea that Mojave Desert tortoises do not exhibit immunological memory. We theorize that the tortoises are actually more reliant upon their innate immune systems as opposed to their acquired. So basically their innate immune systems are just so great at recognizing and eradicating pathogens the first time around that the lymphocytes don't even need to go to their parents, the lymph nodes, to instigate any kind of more specific, stronger response. So some new research has started looking into the B1 lymphocytes of ectotherms and found that they have uniquely phagocytic qualities, meaning that they're able to engulf entire pathogens even only after just that first initial encounter with them. Found that the B1 lymphocytes are able to produce their own natural antibodies, and they also have been measured in much higher levels in ectotherms compared to our own. So this could prove to be a huge factor in why the tortoises' innate immune systems are so much more effective than ours. So these are some of my references and acknowledgements for all the individuals that support this research. Special shout out to the tortoises themselves. And then I just want to thank you all so much for listening to my presentation. Hopefully you learned a little something. And so finally, Poppy, Facilia, and myself want to know if you have any questions. Thanks for listening.